So I'm just here just to talk a little bit about the infrastructure support that we have here in our library. This includes the materials we have, like our whole TRC library, and also a couple of production processes that we have here in place. Now, in our inventory, we have about 300,000 shRNAs. These will target virtually all mouse and human genes. And also, we have a collection of about 50,000 ORF constructs. This is 16,000 distinct ORFs. That's in our entry vector and also a couple of our destination vectors as well. So for our entire inventory, we have both plasmid DNA stocks and bacterial stocks. And also for a small group like the kinase or transcription factor subsets, we have virus that's already been pre-made and also arrayed out. Now, for such a large inventory, we've designed a couple of lab processes that will help you guys utilize the library more efficiently, and we've just streamlined these processes. I'm just going to talk a little bit about our high-throughput plasmid DNA production, our plasmid DNA rearray, and aliquots.
Which the mic to the other side.
Oh. Is that better? A little better? Okay. And then also a little bit about our high-throughput virus production, and briefly mention our pool creation part as well. So we do have a lot of informatics support within our library just to handle the size of this inventory, just from plate tracking, just loading stuff to our database, and also to help the automation process of our lab work. The first part is our high-throughput plasmid DNA production. Even though in our inventory we do have a DNA copy of everything, a lot of our reagents are being requested and used by our screeners. So we are in constant need to replenish these. So everything is done in a 96-well plate format, high throughput manner, and then all the volumes and then information and plate is loaded to our database, which allows us to easily track it and also track volumes for when things are requested out, and also to help us constantly replenish our stocks. So everything from in our SH library and also our ORF library are housed in -80 and -40 degree freezers. This includes master bacterial stocks and also our DNA stocks as well. We always have a master copy that we have archived, and also a couple other copies just as backup in case something was to happen in one of our lab processes. We always like to have backup and safety measures. So all our reagents are stored in these matrix rack tubes, which we give them freezer locations for just so they're easily trackable within our many freezers. And the reagents themselves are stored in these matrix rack tubes that have 2D barcodes on them. And this information will get loaded to the database and is easily accessible and all that. And also by using these matrix rack tubes, it gives us the more flexibility in order to move our reagents around. So when we get a custom request, instead of using the fixed 96-well plates, we can cherry pick out the requested reagents and just form like a new plate just according to our screener's request. So when screeners come, collaborators come, and they're ready to use reagents in our collection, presumably you're working with one of our research scientists in order to actually get these reagents out of our library. But so what actually happens when the clones are requested from our collaborators, and how do we pull the ones that you guys are interested in from such a large inventory, and also ensure that the correct ones were pulled out, that nothing was missed and nothing was, I guess, swapped or wrong? Since we have such a large inventory, we don't want to manually or physically rearray out each tube by hand. It's a very time-consuming process, and it opens up a lot of errors, especially when you're picking out one tube at a time from such a large inventory. So we've used these robotic files that we call Robofiles. These files can be generated either manually, usually on an Excel sheet, or you can use one of our web tools that our informatics team has designed. Now here is just an example of one of our web tools. And this here, you can customize your plate layout, where you can decide which reagents you would like, whether there's a certain order you would like them on the plate, or if you want to completely randomize, you can add in controls or add in any empties you like, and also Within the web tool, it'll tell you what reagent it is, the tube barcode itself, even the volumes that's available, and also give us alerts whether if a reagent is low that we need to go and replenish our stocks before the requests can be fulfilled. So to actually start this whole rearray process, we actually need to go into the freezers and pull out all the source plates and just get them ready just to run them on the robots. So we have all the freezer locations on everything, so it's just going out, pulling out the appropriate source racks, and then just loading them onto the robot to run. And also, to build in some redundancy and to minimize some errors, we have Robofiles that we've generated using the web tools or manually. And also we have software that we use that we can track the destination plates that we can load on, and also the list of source plates that's required for each rearray. So we like a lot of redundancy, a lot of ways that we can automate everything to minimize as much human error as possible. Because if you're pulling out such a huge amount of piles of racks, how do you know that this is the correct one you need? You're not going to be looking at the whole list and say, yes, this is correct. So we like a lot of redundancy built in. And here I can just play a short little video. Nope. Help.
Yep. No, it's loading.
Oh,
It's spinning.
okay. There we go.
There.
So here, when see it's just loading using the software, you can scan in here. You can scan in the deck position, and then scan in your source racks. And then the software here will just keep track of which rack has been loaded and which one still needs to come on. So then,
Oh, that's the Windows guy.
oh, I'm looking at the left. Thanks. So once everything has been loaded, all your destination racks and all your source racks, the robot will read the RoboFile and just cherry pick out the appropriate reagent and just put them in the destination rack just according to the plate map that's been designed. This cuts down on a lot of time spent in the lab, since with this program you can join multiple rearray files at once, so you can run more than one file at a time. So, and also you can see that not the full, everything in the source racks is being used, so this helps minimize the amount of time that if it's bacterial glycerol stocks, that they don't thaw on the bench. There's not a lot of time spent sitting out. So then once everything is cherry picked out, they can just go back into the freezer as quickly as possible. So once the rearray is completed, we also want to ensure that everything that we picked out was correct. So we will just make a scan file, and then this will scan all the 2D barcodes on the rack. And then we'll do a couple of QC checks to make sure that, yes, all the tubes that we wanted did get pulled. There's no random tube in there. The plate layout is correct. The empty wells that you want to be empty are empty. And then if not, we'll get a couple of alerts and then just telling us like where the errors are and to fix them before we go ahead and pass this rearray plate, and also update our database. So once the rearray is done, presumably you would like a copy of your rearray, your reagents. And to do that, we use another robot, so we can just aliquot this more efficiently and minimize human error with like hand pipetting, and also any cross-contamination, since we have filtered tips on the robot as well. Here, you'll see in the video that both the rack tube, the rack destination tube, and the aliquot plate will have barcodes that will get scanned and tracked. Uh-oh. Okay, there we go. So just here, the destination rack is being scanned, the barcode's being scanned, and then this aliquot plate's barcode is being scanned, just so we can track which rack is being aliquoted into which fixed plate. We do a wet dispenser that has water going in first, just so that the amount of DNA has something to dispense into. So here, it mixes a couple of steps before dispensing into the final aliquot plate. So once the aliquots is all set, it's ready to be delivered. We can either deliver the glycerol stalks to our collaborators, or if the library DNA is ready, we can send it over to our virus prep team for virus to be made. Oh, this one's easy. Okay, why am I not seeing?
Better.
Oh, 'cause I can't see it over there. Right?
Yep.
Okay. So our library DNA comes over to our virus prep team now, and they'll take it into our tissue culture facility, where they will process these plates just by batch sizes in a fully automated system. Now, the first part for them is to make up the plasmid, the plasmid mix, and also the transfecting reagent. So here we use a rapid plate, so we can just stamp in all these mixes into the full 96-well plate at once. Everything's fully automated. It's time efficient since you're processing one plate, all 96 samples at once. And here it's stamping in the packaging plasmid and also then the transfecting reagent right into the library DNA. So here all the tips are just stamping in the full 96 samples at once. And then after the transfecting reagent is added, it's incubated for 30 minutes, and then everything gets transferred to the cells. There we go. So once the virus is ready to be harvested, it goes back on the rapid plate where we will pull up all 96 samples at once. We do 2 harvests. So here you can see it's pulling up the second harvest, and it'll mix in with the first batch of virus. And then all the virus will get aliquoted out into 96-well plates. And the number of aliquots just depends on the amount of virus that is requested by our screeners. So here then all the aliquots will get sealed up and then frozen down and delivered. That's about it. Yeah, that's about it. I can stop that. Okay. And that's just a couple of lab processes that we have going on in the lab.